I'll start in just 1 minute. Right, so I'll share my slides. This is the final live session for this year's course of NMR spectroscopy for structural biology. This will mostly be a problem solving session. So in the last 12 live sessions, in each session, we have covered the lectures of that week and we have solved the assignment problems of the previous week. So this week, I will start by solving the assignment of week 12, the last assignment. And then I will go through some practice problems through all the classes from beginning to end. So this is problem 1 of assignment of week 12. Which of the following interactions are absent in isotropic liquids? Now, if you remember the decoupling of the Hamiltonian, so Hamiltonian basically gives you the total energy, and that can be divided into the external and the internal energy. The external energy comes from two contributions. One is the static field, that is the spectrometer frequency that is giving energy to the spins. Another is the RF field, that is the pulse that is applied that gives an additional external perturbation or energy to the spins. So these are the two sources of the external energy. The internal energy has many different subparts. Just 1 second. Right. So as I said, this is mostly a problem solving session. So I'll be solving the problems of assignment 12 first, and then we'll go through some practice problems. So this was the first question of assignment 12, where it was asking that which of the following interactions are absent in isotropic liquids. So if you remember the decoupling of the Hamiltonian, then just 1 second this. Yeah. So you have external and internal energy, and the internal energy has several sources, like the chemical shift. Now, chemical shift is common whether you're taking the NMR in solid state, in an anisotropic liquid, or an isotropic liquid. Then you have J coupling, which is also common in all three, and dipole-dipole long-range coupling, which is basically where the nosy peaks come from, and that is also common in all three. What is specific only for anisotropic liquid or solid is the dipole-dipole short range interactions. You can see it is present in solid and anisotropic liquid, but it is absent in isotropic liquid, and the quadrupolar coupling, which is also absent in isotropic liquids. So if you look at the question, it is asking which of the interactions are absent in isotropic liquids. This third one is the isotropic liquid. It's just cut out at the bottom. So chemical shift anisotropy is present. J coupling is also present. And dipole-dipole long range is also present. So the correct answer is dipole-dipole short range. That is option C. The next question is asking, just 1 second. Why is the solid state NMR spectra of powdered samples broad compared to the solution state NMR spectra? So if you look at the solids, here the two spectra are shown. The solution spectra has very sharp lines, but the solid state has a very broad spectra. This is mainly because of anisotropic interactions. Basically, what happens in a liquid or solution state NMR, you can have spins oriented in many different directions. Like this direction is giving a particular chemical shift. Another orientation is giving another chemical shift, and so on. But in liquids, all these different orientations are rapidly tumbling and averaging out, and you see only one chemical shift that is average of all these. But in solid, the chemical shifts cannot tumble around like that because it is in solid state, and so the orientations are fixed. So each different orientation gives a different peak. And what you see is the envelope of all these peaks, which is why the solid state spectra appears so broad. These are basically called anisotropic interactions, and they are arising because the orientation in the solid state is fixed and the spins are not allowed to tumble and average out. So why the solid state spectra is broad is due to anisotropic interaction. That is the option B. Next is a question. In solid state NMR, sample is rotated at an angle of dash to enhance the resolution as it average out dash. So if you remember the concept of magic angle spinning, so in solid state, the problem that we just discussed in the previous slide, that because the spins are not able to tumble and average out, we need to put the spins in this kind of a rotor and spin the rotor rapidly so that overall the anisotropic interactions average out. So one of these is the dipolar coupling term, which mathematically is equal to 3 cos squared theta minus 1. If we take it to be 0, then the theta needs to be 54.7 degrees. So that is this theta called theta m or magic angle, at which the rotor must be fixed so that when the rotor is rapidly rotated, uh, because the theta is this, the dipolar coupling term will be 0, and the anisotropic interactions arising from this dipolar coupling will also become 0. So this is the value of the rotation angle, and it enhances the resolution. As you can see in the spectra of glycine shown here, glycine in the 13C spectra should have two peaks, one from the C alpha and one from the C prime. Now, as you are increasing the rotation of the rotor, you can see the rotation frequency increases as you go from top to bottom. Slowly, all the anisotropic interactions are averaging out and you are getting a sharper and sharper spectra. Till when you have the rotational speed around 12 kilohertz, two sharp lines are seen as you would expect in the solution state NMR. So this magic angle spinning is enhancing the resolution, and how it is doing that is it is averaging out this dipolar coupling term. So the correct answer is option B, because the sample is rotated at an angle of 54.74 degrees, which was calculated from this equation, and it enhances the resolution as it averages out the dipolar coupling term, which is this. The next question is asking, which of the following process enhances resolution in solid state NMR? So now we will again look back to the concept we saw in the previous lecture. Suppose these green balls are the abundant nuclei like the 1H, and this red sphere is the nuclei with less abundance like 13C. So you can have coupling between 1H 1H, which is homonuclear dipolar coupling, and 1H 13C coupling, that is heteronuclear dipolar coupling. Now if you put the sample in this kind of a rotor and spin it, that is called magic angle spinning or mass, then what it will do is that it will enhance the resolution by weakening these coupling interactions. You can see that both the homonuclear and the heteronuclear dipolar coupling is getting weakened by magic angle spinning. And after that, if you further do RF decoupling, so RF decoupling, what it involves is that selectively the protons are irradiated with a series of decoupling pulses to completely get rid of the heteronuclear coupling. So you can see these red lines here, which were the heteronuclear coupling, are completely getting cancelled out. There is no heteronuclear coupling after this RF decoupling. So both MAS and RF decoupling, both of these are resulting in enhancing the resolution. You can see the pulse sequence here where the excitation starts from 1H and then it is transferred to 13C and you detect the 13C. But the 1H is decoupled using a RF pulse like this on 1H, which results in cancelling out all these heteronuclear couplings. A practical example of this is shown here. The lowest spectra is where you have just taken the solid state NMR without any mass and without decoupling. That is a very broad spectra where nothing can be resolved. This spectra is where magic angle spinning has been done, but the RF decoupling has not yet been done. So you can see the resolution has increased, but it is still not the sharp line that is expected from the solution state NMR. And finally, the topmost spectra is where both magic angle spinning and the RF decoupling has been done. And this gives very sharp lines. And now the solid state NMR is comparable to the solution state NMR. So in order to enhance resolution in solid state NMR, both magic angle spinning, that is option A, and heteronuclear decoupling, that is option B, is done. So the correct answer is D, that is both A and B. We will go to the next question now, that is asking, in 2D PDSD or DAR, proton is excited with 90-degree pulse. If the duration of the pulse is 5 microsecond, what is the power being applied on the proton? So now we will look back to this pulse sequence. Basically, in the pulse sequence, it starts with a 90-degree pulse on proton. After this 90-degree pulse, there is a cross-polarization sequence, and that results in transfer of the magnetization from proton to 13C. Then a certain time T1 is given for the magnetization to evolve in 13C. And then there are more pulses and then a mixing time. We don't need to look in the details of that. What you need to remember is this formula. That is, power is 1 by 4 times duration of the pulse. An example is shown here. This first 90-degree pulse on 1H, the duration is 2.5 microsecond. So power would be 1 divided by 4 into 2.5. And because it's microsecond, in denominator you will have 10 to the power minus 6, which will go up in the numerator as 10 to the power 6. So if you calculate this, you will get, just one second, I'll just, in order to show this, so if you calculate this, you will get 100 kilohertz, and you can see 100 kilohertz is the power that is applied here. So a 2.5 microsecond pulse, if you apply this formula, will give you a power of 100 kilohertz. So if we apply that to our question, it is saying a pulse of duration 5 microsecond. So power would be 1 divided by 4 times duration of the pulse, that is 5 into 10 to the power minus 6. If you calculate this, it comes to 50 kilohertz. That is your option B. So option B is the correct answer. We move on to the next question, which is saying, which of the following method enhances sensitivity in solid state NMR? So this, is done by cross polarization. Basically, the cross polarization technique is where you first excite in using a 90-degree pulse in the 1H channel, and then just one second. Right, so I was discussing this question. So which of the following methods enhance sensitivity in solid state NMR? So we know that cross polarization is used to increase the sensitivity. And how that is done? First, this 90-degree pulse is applied on 1H, and then there is a certain pulse sequence called cross polarization, which basically transfers the magnetization from 1H to 13C. Now, what is the advantage of this is that the gyromagnetic ratio of hydrogen is 4 times that of carbon. So because we are detecting the FID on carbon, we will get a very weak signal. The sensitivity will be very low. So if we want to increase the sensitivity of this FID, we have to do cross polarization so that the magnetization is transferred from hydrogen to carbon, and this higher sensitivity of hydrogen is taken up by carbon. So that is what happens through cross polarization. You can see an example of two spectra. So there is a mixture of two compounds. If you just do direct polarization, direct means that you have excited on 13C and you're collecting the FID on 13C. Basically what we were doing for solution state NMR so far. Then a very broad spectra is found without any resolution. But if this cross polarization pulse sequence is done, where you first excite in one edge and then allow the magnetization to transfer to 13C, so that the 13C detection becomes more sensitive, then you can see clearly very sharp peaks appear, and you can even distinguish that which are the peaks of compound 1, which are the peaks of compound 2. So cross polarization is a method that is used to increase the sensitivity in solid state NMR. So the correct answer is option A. Then you have this question that is saying dash base transfer is used to study mobile part of amyloid fibrils, while dash base transfer is used for studying structured static part. So again, look, going back to the previous class, as I said, I'll first cover the assignment problems of the previous class, and then I will discuss some practice problems from the beginning to the end of all the classes. So in the previous class, we covered something called dynamics-based spectral editing. So basically, suppose you have a protein where you have a certain part that is folded, and certain part that is unfolded. So this is the static part of your protein and this is the mobile part of your protein. So in order to study the mobile part of the protein, we can do J coupling based experiments, basically what we were doing for solution state NMR, like inept or inteptopsy, in order to find out which amino acids are involved in this mobile part of the protein. But the static part of the protein will need solid state NMR. And for that, what is done is dipolar based transfer of magnetization. Because we know, we just saw a few slides ago that in solid state NMR, the transfer of magnetization mainly happens through dipolar coupling, which is why when we were doing the magic angle spinning, the dipolar coupling term had to be decoupled and made zero. So for the static part of the protein, dipolar based experiments are done, like PDSD or DAR, in order to study the static part of the protein. So doing this, we can find out that which are the amino acids that are aggregating in the static part and which are the amino acids that are in the mobile part. So the question says, for the mobile part of the fibril, we basically J coupling is done, that is option A, while for the static part, the magnetization is transferred by dipolar coupling, that is also in option A. So the correct answer is option A. The next question is asking, which of the following solid state NMR experiments is equivalent to the liquid state 3D experiment HNCACB? So if we compare the two, in liquid state, the HNCACB experiment, basically the magnetization starts. So here, this is I minus 1, this is I. So in HNCACB, the magnetization starts from the H of the I residue. It is transferred to the N of the I residue. And from N, it goes to C alpha, both the C alpha I and C alpha I minus 1. And from the C alpha, it can then go to C beta I minus 1 and C beta I. This was the case in liquid state NMR. But we know in solid state NMR, we cannot use hydrogen because hydrogen gives very broad spectra, and so the resolution is not good. So in solid state NMR, the equivalent experiment is We start from again from the amide nitrogen, but this time we don't detect the amide nitrogen. Here again, this is your I residue and this is your I-1 residue. So in solid state NMR, the magnetization is transferred from N again to C alpha, and from C alpha it can go either to C beta or it can go to CO. So here the transfer of magnetization is slightly different. But so how can we say that these are equivalent? Mainly what we look at when we are considering equivalent is whether it is we are detecting the same kind of atoms. So what are the atoms we are detecting here? The ones marked in red. We are detecting H, N, and both the C betas here. Here what are we detecting? Again, we are not detecting H, because in solid state we can't detect H, but we are detecting N and the C beta. So that is how you can say these two are equivalent. So if we look at the options here, this is the closest that is there, NCACX. Because if you imagine C beta as CX, it will become NCACX. And we don't detect hydrogen because it's a solid state experiment. So NCACX is the correct answer. That is option B. The ninth question of this assignment was asking, just one second, I'm not sure if so. The ninth question of this assignment was asking, which of the following statement is true regarding membrane proteins? So we saw a lot about membrane proteins. So we know membrane proteins are ones that are embedded inside the membrane. So the problem with these membrane proteins is that they are not soluble. And if they are not soluble, you cannot do solution state NMR. So we have to do solid state NMR. And also they are very difficult to crystallize. So you cannot do X-ray crystallography also. So solid state NMR remains the only technique to study these membrane proteins. But one problem is that if we isolate the membrane protein outside the membrane, they will lose that structure. They will adopt some other structure. And that is not of any use because we want to study the structure in the native environment. So the thing that we have to do the solid state NMR of these membrane proteins is we have to mimic the membrane environment. For that, what is done is different lipid or detergent molecules are used to form micelles or liposomes around the membrane protein so that the native membrane environment can be mimicked. So depending on what kind of detergent molecule you used, the type of membrane is different. Like if you use DOPC, you can see these detergent molecules are slightly longer than DMPC. Or if you use DOPE, these molecules are putting a pressure on the protein. You can see these are bending inwards and putting a pressure on the protein. So that will change the protein structure. That is what was observed even through the solid state NMR. So for DOPC, the solid state NMR was taken, and for DOPE, the solid state NMR was taken. And it was seen that they were giving two different conformations. So if we look at option A, it is saying membrane protein can adopt different conformation depending on the lipid membrane in which they are embedded. That is true because that is what we saw, that depending on the lipid used to make the membrane, the protein gave a different conformation. So option A is correct. Then option B, membrane proteins are highly soluble and easy to crystallize. This is wrong. Because they are very insoluble and because they are difficult to crystallize is why we have to use solid state NMR in order to find their conformation. Then C, which is saying there is no effect of lipid membrane on membrane protein conformation. That is also wrong. We just saw that lipid membrane, what we are choosing as the lipid is affecting the conformation. So correct answer is option A. And this was the last question of assignment 12, which was saying, if we are spinning a solid sample at 20 kilohertz, and frequency applied on carbon nuclei is 80 kilohertz. In order to meet the cross polarization condition, what can be the possible value of frequency applied on proton? So again, we will look back to this pulse sequence that was there. So here, if you look at the region of cross polarization, the frequency applied on one edge was 67.5. Frequency applied on 13C was 50 kilohertz. Now for condition of cross polarization, this is the equation to remember, that omega r is omega h minus omega c, where omega r is the spinning frequency. So that is given by omega h, which was 67.5, minus omega c, that was 50, which gave an omega r of 17.5 kilohertz. So this is the spinning frequency of the rotor that is spinning at the magic angle. This is the frequency that is applied during mixing time, because this is the time where you're mixing all the magnetizations to cancel out the anisotropic interactions. So 17.5 is what was applied at the mixing time. For our question, we'll again use the same formula. Omega h minus omega c is omega r. What is given in the question? Omega r is given. So this 20 kilohertz is the spinning frequency, is omega r. And frequency on carbon nuclei is given. This is omega c. So in this equation, you know omega c, you know omega r. You just have to rearrange it and find omega h is 100 kilohertz. 100 kilohertz is your option D. So the correct answer is option D. So as long as this condition is fulfilled, that is, the spinning frequency is the difference of frequency of hydrogen and carbon, then the cross polarization condition is fulfilled, and whatever FID you detect on carbon, it will be a very well resolved spectra. So this basically covers the questions of assignment 12. Now, since this is the final class, I will go through some practice problems covering many concepts from the first class to the end. So let's go through these questions. One question is, re-establishing the population difference between higher and lower energy spin levels is dependent on what? We know that in NMR spectroscopy, you will have two levels, alpha and beta. And when you apply an RF pulse, the population changes due to excitation from alpha to beta. But after the RF pulse is stopped, the population difference again goes back to the equilibrium. And how does this happen? So that is the question. So the answer is that this happens through spin lattice relaxation. So I will, how I will do this class is I will solve the question and then I will briefly discuss the concept also. So you will kind of get a revision of the concepts also. So we will just look at spin lattice relaxation. So in NMR, you have two levels, the lower for spin half, and which is the lower level and the upper level. And there is a population difference between the upper and lower level that is defined by the Boltzmann distribution. So the population in upper divided by population in lower is e to the power minus delta E by kT, where delta E is this energy difference. And we know delta E we can write as h nu. And for NMR, we can write the energy difference between the two levels as minus gamma h bar B0. So for NMR, N upper by N lower becomes e to the power minus gamma h bar B0 by kT. So this is the population difference at equilibrium. Now, if we add an energy is given, an RF pulse or energy is given, spins can be promoted to the upper level or they can come down to the lower level. Overall, basically, the population will adjust. Now, after the pulse is stopped, the population has to go back to equilibrium. And how this happens is spin lattice relaxation. So lattice is basically everything other than the spin system you are studying. So after the pulse is stopped, the spins in the upper level will come down, and while this happens, an energy will be released. Where this energy will go, this will be absorbed by the lattice in order to promote its spin from lower to higher. Similarly, if a spin is promoted from lower to higher, it has to absorb some energy. And where this energy will come from? It will come from a spin in the lattice that is coming down. So when the lattice spin comes to the lower energy, it will release an energy, and this will be absorbed by the spin. So basically, whether uh, uh, the energy is released or absorbed, it is done from the lattice, and that is why it's called spin lattice relaxation. So if the population, there is a equation that defines the population difference. So this population difference at equilibrium, if we consider it as n0, and the population difference at any time t is n, then this equation is there that n is n0 into 1 minus e to the power minus t by t1, where T1 is the spin lattice relaxation time, which is also known as the longitudinal relaxation time, because if you kind of imagine the spin going up and down, that is why it's called the longitudinal relaxation time. So uh, you can see that if the T is infinity in this equation, the population difference is the equilibrium population difference. So what this means is that as the time goes, the population difference will slow. So as long as there is a population difference, you will get an NMR signal. But the moment the populations become equal, there is no net absorption or release of energy. The NMR signal will completely uh, vanish. So that is what spin lattice relaxation does. And if we go back to the question, you can see it again now that re-establish, just 1 second. Right. So that is why the answer for this is spin lattice relaxation, because that is what is helping to re-establish the population difference between the alpha and beta states. The next question is asking, more the number of electron withdrawing groups near the proton, what is the effect on the chemical shift? So again, we will look into the concept. So the nucleus is surrounded by electrons. One field is the external magnetic field you're applying, that is B0, which is upwards. This magnetic field is causing the electrons to move, and the movement of charged particles or electrons creates another magnetic field called the induced magnetic field, which is in the opposite direction. You can see it is downwards. So the induced magnetic field reduces the net magnetic field uh, that is affecting the nucleus. And so you can see here, if you have many electrons surrounding the nucleus, the induced magnetic field will be, if you have lowered electron density, so less number of electrons surrounding the nucleus, then the net magnetic field will be stronger, because the let's see, net magnetic field, let us consider that as the original magnetic field of the spectrometer B0 minus the induced magnetic field. So when electron density is low, induced magnetic field is low. So net magnetic field is high. Or if the electron density is high, then induced magnetic field is high. That makes the net magnetic field low. This is what is affecting the chemical shift. So now we will look at how this electronegativity is affecting. So suppose you have carbon, hydrogen, and an electronegative element like chlorine attached. So chlorine is an electron withdrawing group. It will withdraw electrons away from the proton. So proton will get a delta plus charge. That is, it will be this situation where the electron density is lower. And when electron density is lower, we know that it moves at higher ppm values, that it moves downfield. So the chemical shift becomes greater. So now if we go back to the question, what happens to the chemical shift? More the number of electron withdrawing groups. We will look at this example. You can see this example. CHCl3, there are 3 chlorines near this underlined proton. CH2Cl2, there are 2 chlorine. CH3Cl, there is 1 chlorine. So as the number of chlorine is increasing from 1 to 2 to 3, you can see the chemical shift value is also increasing from 1 to 2 to 3. So more the number of electron withdrawing groups, higher the chemical shift. So the correct answer is C. The next question is asking, the frequency range of chemical shift of proton in a 700 megahertz spectrometer is how much? So we will look at the formula now. So chemical shift delta I is defined as HR minus HI by HR into 10 to the power 6, where HR is the reference and HI is the field by the specific nucleus. This can be converted in terms of frequency. You can write it as nu I minus nu R by nu 0, where nu I is the frequency of the particular nucleus you are studying. Nu R is the reference frequency. We know that in NMR, normally a reference like tetramethylsilane is taken, which gives ppm values 0. And nu 0 is the spectrometer frequency. So once we know this, now we will apply what is given in the question. The question says that the spectrometer is 700 megahertz. So this is your nu 0. So nu 0 is given as 700 megahertz in the question. So we will put it in this formula. Delta is nu I minus nu R divided by nu 0, that is 700 megahertz into 10 to the power 6. We can assume the delta as around 10, because we know the range of chemical shift, the delta values of proton ranges from around 1 to 10. So we take it around 10 and we define delta nu as nu I minus nu r. So delta nu I minus nu r, if we move the 700 to this side, is delta into 700 megahertz divided by 10 to the power 6. Delta nu comes out to be 7 into 10 to the power minus 3 megahertz, or you can write it as 7 kilohertz. This is the value of delta nu. That is the frequency range. So we got 7 kilohertz. So basically that falls in option C, that is 5 to 10 kilohertz, option C. And so option C is the correct answer. The next question is asking, radio frequency induced transition probability is proportional to what? So if we look at briefly into the kinetics of NMR, we know that when spin is half and the field is off, there is only one energy level. But when field is switched on, the lower energy level is when the spin is in the same direction as the B0. Upper energy level is when the spin is in opposite direction of B0. Now, when you apply an RF pulse, it can cause an upward or a downward transition with equal probability. And we will call this probability of transition as P. Now, if we look at the kinetics, so if we imagine it as a first order rate equation, so you have alpha spin going to beta spin and beta spin coming back to alpha spin. So if you imagine this as a first order rate equation and putting the first order reversible rate equation kinetics, you will get that the population difference n at time t is the population difference at equilibrium n0 into e to the power minus 2pt, where p is this RF induced transition probability and t is the time. This transition probability is defined by this equation, which is 1/4 into gamma squared, which is the gyromagnetic constant squared, into h1 squared, that is the RF power that is applied. So the probability of transition is dependent on the square of the gyromagnetic constant and the square of the RF amplitude. So now if we look back, the radio frequency induced transition probability P is proportional to the square of the gyromagnetic constant and the square of the amplitude of the radio frequency. So it is both A and B, so C is the correct answer. We will move to the next question. This is saying in methyl salicylate, the chemical shift of the OH protons is 14 ppm, which is much higher than that observed in alcohols. Normally in alcohols, you will see a shift of 5 to 7 ppm. So why is the alcohol proton of methyl salicylate, this compound, so much more downfield shifted? We will look into the concept that is hydrogen bonding. Now, hydrogen bonding basically occurs when there's a hydrogen attached to an oxygen, fluorine, or nitrogen with another such hydrogen. So we know that the oxygen will have a delta minus charge, the hydrogen has a delta plus charge, and again the other oxygen has a delta minus charge. So this is how the hydrogen bonding occurs, and what happens is because of the two oxygen on both sides, more electron density is withdrawn away from the hydrogen and this delta plus increases. So if a hydrogen is involved in hydrogen bonding, it will go more and more downfield. Now, alcohols generally have chemical shifts if it's a free alcohol from 0.5 ppm to if it has a lot of hydrogen bonding 5 ppm. But methyl salicylate has an abnormally high of 14 ppm. That is because there is an intramolecular hydrogen bonding. You can see within the same molecule, the OH and the C double bond O at the ortho position is forming a hydrogen bond. Now, intramolecular hydrogen bondings are much stronger, and especially because it is forming a stable 6-membered ring, this is an even more stronger hydrogen bond. Because the hydrogen bond is so strong, the delta plus of this hydrogen increases to very high extents. And because it is increasing to such high extents, the methyl salicylate hydrogen is highly downfield shifted to around 14 ppm. So the correct answer is C, that is intramolecular hydrogen bonding. We will move to the next question. So it is asking, the folding in signal in NMR spectrum can be prevented by what? We will look into a few features of the NMR spectra. So there is something called a single channel versus a quadrature detection. In single channel detection, what happens? Suppose this is your spectral range. Spectral range is from here to here, and your carrier frequency shown by this black arrow is at one end of the spectral range. So all the other frequencies are in the positive direction of this carrier frequency. So only one channel detection is fine, because if you just have a channel in the positive direction, you will be able to cover the entire frequency range. This is called single channel detection. If the carrier frequency is somewhere in the middle of your spectral range, then you will need two channels, one in the positive direction, one in the negative direction. And this is called quadrature detection. Now what happens is that, suppose you have a spectral width shown by this arrow, and you have the carrier at one end of the spectral width, that is single channel detection. But the signal that you have is outside your spectral width. What will happen in this case is that an error will result, which is called a folded signal. So you can imagine this signal will, if you imagine this as a paper that is getting folded from here, this signal will come here, but it will have an opposite phase. So this is called a distorted phase or a folded signal. Similarly, for quadrature detection, you have a spectral width of this much, with the carrier being at the middle of this spectral width. Now, and also a frequency lying outside your spectral width. Now, if you do the quadrature detection, again, this will kind of be folded inside your spectral width, but it will have an opposite phase. You can see the phase is again distorted. So this folding, when is it happening? It happens when you have a frequency that is outside the range of your spectral width. So how this can be prevented is that you increase the spectral width. So if the spectral width was increased to this much, then this frequency is falling inside your spectral width, and then this will not happen. So the way to prevent folding of signals is by increasing the spectral width, that is option A. We move to the next question, that is asking, according to the Nyquist theorem, what is the minimum number of points that must be sampled per wave to get a proper FID? So we will again look at some parameters and criteria of the NMR. One is the signal digitization and sampling. Basically, if you have an FID, many points along the FID are collected in a digital manner. And we call the time between two consecutive points as a dwell time. So say the time between this point and this point. This time is called the dwell time or dw. Nyquist theorem says that there must be at least 2 points per wave to represent the frequency correctly. So if you have multiple waves, for each wave, so you can see here, one wave is shown in purple, one wave is shown in blue. We must have at least 2 points per wave to represent the frequency correctly. So that is your Nyquist theorem. That is what this question is asking. So the minimum number of points sampled per wave must be 2, that is option C. Now, once we collect this FID by sampling at least 2 points per wave, now suppose there is an error. We stop collecting the FID before it fully decays. You can see the FID is decaying, but before it has fully decayed, we have collected it. Then what will happen? There will be some distortions in the baseline of the FID after we do the Fourier transform. In order to prevent this, there are certain techniques. One is called digital filtering. So suppose we are truncating the FID here, that is before it is fully going to 0, then we will get distortions in the baseline. In that case, we have to multiply the FID with a function that goes to 0 at the last data point, that is the point of truncation. That is, this is our last data point. We have not collected these. So we must multiply it by a function that goes to 0 here. And then when the Fourier transform is done, we will get a clean spectra with no distortions at the baseline. Another way is that we have collected a truncated FID here. Suppose we have collected n points. The way is to collect an additional n points after. So we have collected up to here. We have collected n points. If we collect an additional n points after this and we set all the points at 0. So that will increase the total number of points to 2n, and that will improve the resolution of the spectra. So these two methods, digital filtering and zero filling, both improve the resolution of the spectra. So these are certain concepts. As I said, I'll not only be discussing the questions, but I'll also be discussing the concepts. So now there is a question asking about the inept pulse sequence. We will look at the, first we will look at a concept called selective population inversion, and then we will go to inept. So in selective population inversion, so we'll start with a two spin system, A and X. So you can see there are 4 energy levels, and you can there are 2 spin systems, so they will couple with each other. We know that when there are 2 spin systems, they will couple with each other, and we are making one assumption that capital delta is greater than small delta. So there will be 4 energy levels. Delta plus delta is the lowest. Delta minus, this is basically, so you have 2 spins. Lowest P0 is in the upwards direction. The lowest energy is when both the spins are in the upwards direction, which is delta plus delta. Highest energy is when both the spins are in the downwards direction, minus delta minus delta. The other options is that one is up, one is down, or one is down, one is up. These are the 4 options, 4 energy levels that you have. So now the possible transitions, we know only single quantum transitions are possible. So it can only go from lower energy. So these 2 red lines shown are possible. These are, so X will be split into 2 lines by A, X1 and X2. And A will be split into 2 lines by X, that is A1 and A2. And the intensity of these lines is proportional to the population difference between the states. So A1 is this transition. And if this is delta plus delta is the population. If we subtract the population, we will get 2 delta. So the intensity is proportional to 2 delta. Similarly for A2. And for X1 and X2, we can see that the intensity is population to 2 small delta. The problem of this is that the X1, X2 the signal is low. So in order to cause signal enhancement, a technique called selective population inversion or SPI is done. In SPI, we are selectively inverting the population of 2 levels. So this level, that is minus delta plus delta, comes down, minus delta plus delta. And this level, that is delta plus delta, comes here. So these 2 levels have exchanged their populations. Now, if you again calculate the population difference of the A1, A2 and X1, X2 transitions, you will see that A1, A2, the intensity remains the same, that is 2 capital delta, but the transitions become anti-phase. So here they were both in phase, same direction. These are now in opposite phase, anti-phase. But the advantage is that the X1, X2 signals, which were weak, they have now significantly increased in intensity. So that is the advantage of selective population inversion. It helps in increase in intensity of the weak signal. But there is disadvantage also. Disadvantage is now that the signals have become anti-phase. We need the signals of the NMR spectra to be in the same phase, but they have become in opposite phase. So this is the disadvantage of the selective population inversion. In order to combat this, uh, or uh, uh, and get more, in order to combat this and get better spectra, this technique was introduced called INEPT. INEPT stands for Insensitive Nuclei Enhancement by Polarization Transfer. So in the initial part of INEPT, so suppose the 2 spin is A and X. Initially, a 90x pulse is applied to A. So both A1 and A2, they were initially in Z. Because of the 90x pulse, they come to Y. Then a certain time tau, which is equal to 1 by 4j, is given in order to allow the A1 and A2 to move by an angle of theta. So this theta will be equal to 2 pi j into tau, and we keep this as 90 degree. So we set the time tau in such a way that the angle between A1 and A2 becomes 90 degrees. After this, a 180x pulse is applied on A, and what this will do, this will flip A1 to this side and it will also flip A2 to this side. So now A1 and A2 are in the opposite direction. The 180-degree pulse is simultaneously also applied on X, the X channel. So 180-degree pulse is also applied on X simultaneously, but this will not affect the A spins. Or rather, it will not affect the magnitude of the A spins, but the direction of the A spin is changing. You can see after the 180x is applied, before the 180x was applied, A1 was traveling in this direction. Now A1 is inverted. And initially A2 was traveling in this direction. Now A2 is inverted. So the 180 pulse applied on A channel is moving the pulse completely by 180 degree. And the 180 pulse applied in the X channel is just changing the direction of rotation of the pulse. After this, another time period tau is given, in which, so we know in time period tau, the spins move by 90 degree. They are already separated by 90. If another time tau is given, they will now be separated by 180 degrees. And finally, a 90-degree pulse is first applied on A. And if we are applying the 90-degree pulse on A, both of these, they are in X, they will go back to the Z axis. We know detection is always along the Z axis, so we need the magnetization to come back to the Z axis. And finally, a 90 pulse is also applied on X and the FID is collected. So when this 90 pulse is applied on X, the X magnetizations, so X magnetizations initially they were in the Z direction, but you need them also to come in the XY plane, and so that is what this final 90 degree pulse is doing. So this is the entire sequence of the inept experiment, and what this is doing is that here after SPI, the population, the intensity of the two peaks X1 and X2 were different. One was -2 delta plus 2 small delta, another was 2 delta plus 2 small delta. So these are different frequencies. And what this inept experiment is doing is at the end of the inept experiment, the X1 and X2 have same intensities. It is 2 delta and -2 delta. So that discrepancy is removed. Now, so inept experiment is kind of extension of the selective population inversion. At the end of selective population inversion, the both the intensities of A and X were increased, and the A spins A1 and A2 had equal intensity but opposite direction, but the X spins had unequal intensity. At the end of inept, the X spins also have equal intensities, and that is the advantage. And so this is the inept pulse sequence. If we go back to the question, the inept pulse sequence starts with 90x. So automatically option A is cancelled out because it is 180x. So 90x applied on A, all three are saying that, then time tau. B and C are saying time tau, but D is saying time tau by 2. This is wrong. This gets cancelled out. After the time tau, it's 180x applied on A and 180x applied on X. 180x applied on A and X. B is saying that, but C is saying it wrong. So the correct answer is B. Let us just continue along. After the 180x pulse applied on A and X, there's a time tau. Then there is a 90y pulse applied on A and a 90x pulse applied on X, after which the FID is acquired. So B follows this entire pulse sequence. So B is the correct answer. Now we have a question saying a proton exchanges between two sites A and B. The proton chemical shift difference between the two sites is 1000 Hertz. What happens at the coalescence point? So if we look back to dynamic NMR, so suppose we have two protons A and B and they are exchanging with each other. We can call the rate of exchange Kx as the sum of the rate of the forward direction reaction K1 plus the rate of the reverse reaction K minus 1. And K1 can be written as inverse of tau A, that is the lifetime in the A state, and K minus 1 can be written as inverse of the lifetime in the B state. So we can write K exchange as K1 plus K minus 1, which can be written as 1 by tau A plus 1 by tau B. And we will call 1 by tau A plus 1 by tau B as 1 by tau. So this is how we define tau. Now, as the rate increases, so initially, suppose the exchange rate is very slow compared to the difference in frequency between the two states. So we can see the difference in frequency is around 250 hertz, the difference in this frequency. So if the difference in frequency is around 250 hertz and the K exchange is much smaller, it's only 10 hertz. So you will see two distinct peaks. Now, as the temperature increases, as we go from low to high, temperature is increasing. rate of exchange is also increasing. The two signals start merging. There is a point called coalescence point where you cannot distinguish the two signals anymore. At that point, there is a certain criteria. The criteria is that tau, that is 1 by k exchange, into omega A minus omega B, this frequency difference, is 1.414. This is the definition of the coalescence point. And if we further increase the temperature to a point where the exchange rate is much greater than this difference in frequency of the peaks, then only one peak will be seen. So this equation is important to remember for the coalescence point. If we go to the question, it is saying at the coalescence point, what happens? Option A is saying exchange rate is much smaller than the difference in frequency. But if exchange rate is much smaller, you will see two separate lines like this. So this is not coalescence. This is wrong. Exchange rate is much larger than the difference in frequency. Then you will see just one peak. This is also not coalescence. Coalescence is something like where the two peaks have merged, but they have not become one sharp peak. There it is this criteria we just saw, that tau into omega A minus omega B is 1.414, where tau is 1 by exchange rate. So this is the correct answer. So the correct answer is option C. Just give me 2 minutes. Now the question is asking the relation between NOE and distance between spins is what? So we know that NOE is basically polarization transfer, that is the transfer of magnetization from one spin to another. And it stands for nuclear Overhauser effect. Now, NOE, what we do, suppose we have a compound with many spins near each other. We are perturbing a specific spin. We are applying a specific spin at HA. Now, this polarization will transfer to the spins that are close to HA in space. So from HA, it can transfer to HC or it can transfer to HB. And if we allow even more time for mixing, from HC, it can go to HD, HB, it can go somewhere further, and so on. So this happens by dipolar coupling between the protons. So using this, you can find out which are the spins that are close to each other in space. And the intensity of the NOE is directly proportional to 1 by r to the power 6, where r is the distance between the spins of which we are observing the NOE peak. So if we look at the options, the correct answer is D. That is, NOE is directly proportional to 1 by r to the power 6. The next question is asking, in the multi-pulse 2D experiment, which of the terms are not directly observable? So if you remember, the quantum condition is that delta M must be plus minus 1. That is single quantum transition. So if you have two spins of spin half, you have 4 energy levels. We have already discussed this. Now, transition from alpha alpha to alpha beta or beta alpha is allowed because M is changing from 1 to 0. So delta M is 1. Or from alpha beta to beta beta or beta alpha to beta alpha beta is also allowed because from 0 it is becoming minus 1. So these red arrows that are shown are allowed because these are single quantum transitions. But if you go from alpha alpha to beta beta, this is double quantum coherence because delta M is 2. And this is not allowed. And also between alpha beta and beta alpha, the delta M is 0. So it's called zero quantum coherence. This is also not allowed. In terms of magnetization, how this is indicated is that if you have single quantum coherence, there will be just one I term, like if it is IKX, that indicates single quantum condition. But if you have two terms like 2 ikx, iky, that indicates a mixture of double quantum and zero quantum coherences. So if you have terms like 2 iky, ilx, 2 iky, ily, these are not directly observable because in the flow of magnetization, you can only observe single quantum coherence. Single quantum coherence are terms like ikx. So containing a single magnetization. You can also have sum or difference of these terms. These terms that you see here, these can be added up or they can be subtracted, but they will eventually to give you pure double quantum coherence or pure zero quantum coherence. But none of these can be detected. Only the single quantum coherence can be detected. So the question is saying, so if you see, this is your, either it is double quantum or it is zero quantum. Same for this. So none of these are directly observable. So the correct answer is both A and B because you cannot observe A or you cannot observe B directly. So the correct answer is C. The next question is saying, which of the following correlations is not observed in 2D cozy spectrum of a proton? So in cozy spectrum, what happens is that there is 3-bond coupling. So from NH, NH-C alpha correlation can be observed because there are 3 bonds, 1, 2, and 3. So NH-C alpha correlation, you can see NH-C alpha will give you this peak. Similarly, C alpha, C beta, C alpha and beta is giving you, H alpha, H beta is giving you this peak. Then H beta, H gamma is giving you peaks. But you will not see any peak between H gamma and H gamma because one H gamma is here, one H gamma is here. We'll call it gamma 1, gamma 2. And how many bonds are there in between? 1, 2, 3, and 4. So 4-bond coupling is very small and you cannot observe it on the cozy spectra. So cozy spectra will only give you 3-bond couplings. And that is what is the question asking. So which of the correlation is not observed? We just saw NH-H alpha, you can see. H alpha, H beta, you can see. Beta gamma, you can see. But gamma 1, gamma 2, you cannot see. And that is your answer. That is D. Next, it is asking about the fine structure of a cozy cross peak in a linear 3-spin system. So suppose we are looking at a cozy cross peak. Now, if we are, we are looking at the AM cross peak. So if we directly take, so let us first imagine a linear system AMX, 3 spins connected in a linear way. Now, if we are looking at the AM cross peak, AM is called the active coupling and it will always cause anti-phase splitting, meaning into plus minus. But MX, X is not part of this cross peak. That is why it's called a passive coupling. So there will be no sign change. Plus will split into plus, minus will split into minus. Now let us look at how this fine structure comes. We start from the frequency of A. A will couple with M. So JAM is the active coupling. So it will give two different signs, plus and minus. And M will couple with, so A will couple with M, but A cannot couple with X. It is too far. We only consider adjacent couplings because this is cozy. So, but M, it can couple with both A and M can couple with X. So M will first couple with A And because AM is the active coupling, it will split into plus minus. Then M will couple with X. MX is the passive coupling. So there is no sign change. Plus will split into plus plus, minus will split into minus minus. So now we have M on the X axis, M meaning plus plus minus minus, so plus plus minus minus. And we have A on the Y axis, that is plus minus, plus minus. So this is how the cross peak is splitting. And one thing to note is that this will only happen if AM is greater than MX. So you can see MX, they are not overlapping. But what if AM is less than MX? In that case, these two will overlap. You can see the condition here. M being split by A is giving you plus minus similar. But now if AM is less than MX, then these two will overlap. This will come here, this will come here. Then it will become plus minus plus minus. That is what is here. So the fine structure will change depending on the relative magnitude of AM and MX. So that is what this question is asking. In a linear 3-spin system, you cannot predict the fine structure. You can see C is the correct answer, because if AM is greater than MX, that is this first, it is plus plus minus minus. That is option C. And if AM is less than MX, it is plus minus plus minus. AM is less than MX, it's plus minus plus minus. So the correct answer is option C. So depending on the relative magnitude of the active and passive coupling, you will get the fine structure of the cozy cross peak. Next, the question is asking, which of the following 3D NMR experiment can be used to detect the side chain of the amino acid in a polypeptide chain? So if we look at the different 3D NMR experiments, first option said HNCO. HNCO, you start from the amide hydrogen, magnetization is transferred to nitrogen, and from nitrogen it goes to the carbonyl. So it does not go to the side chain of the amino acid, it is only in the backbone. So HNCO is cancelled out. Then what about HNCA? In HNCA, you start from the hydrogen, transfer the magnetization to nitrogen, and from nitrogen it goes to the C alpha of both I and I minus 1. So again, you are not going into side chain, you are only staying in the backbone atoms. So H and Ca is also cancelled out. Then it is HNCaCO, that is this HNCaCO, that is this fourth option. Fourth option, this is kind of like extension of HNCa. In HNCa, you have the magnetization going from H to N and from N to the 2C alpha on two sides. Here you have the magnetization going one step further. From C alpha, it goes back to CO. And here also you can see alpha, it goes to the next CO. So here also magnetization is transferring only within the backbone. So C is also cancelled out. And finally, you have HNCaCb, that is this one, where magnetization goes from H to N to C alpha on both sides, and from C alpha it goes to C beta. So you are entering C beta, that means you're finally detecting the side chain of the amino acid. So using this technique, you can determine that what is the amino acid, because unless you know the side chain, you cannot know the amino acid. So HNCaCb helps you detect the side chain. So the correct answer is HNCaCb, that is option D. Then you have a question saying, in the HNN experiment, the peak in the plane, so you have a plane which will show you the F3, F2 plane. So what is the sign of the residue at I minus 1? So basically it is saying you have a tripeptide, glycine X, Y. So you know that the HNN experiment detects 3 consecutive amino acid residues, and the question is telling that the residue at I-1, this position is glycine. So what will be the pattern of the peaks? So we can see it is saying the F3, F2 plane. So it is talking about, it is talking about the plane, and you can see that what it should be, that, sorry, it is talking about the F1, F3 plane. So the uh, information given is that glycine is at I-1. Glycine I-1 refers to this. And we know that this dark color means that it is positive. So glycine I-1 means the pattern is plus, plus, minus. That is your plus, plus, minus. That is your option C. So what you can remember is that the general case is, this is the general case where you have plus, minus, minus. So this is the general case. Now, whenever you have proline, one peak will be missing for each proline because proline does not have a amide hydrogen. And whenever you have glycine, the pattern of peak will change. You can see if glycine in the middle, it becomes minus, minus, plus. Glycine at I-1, what we just saw is plus, plus, minus. Glycine at I+1 is minus, plus, minus. So wherever you have glycine, the sign will change, and this is why glycine acts kind of like a checkpoint. So here the correct answer is option C. Now there is a question asking, guanine binds to uracil in what kind of base pairing? We know that in normal Watson-Crick base pairing, this is the general Watson-Crick, A will bind to U or T through 2 hydrogen bonds. You can see this is one, 2 hydrogen bonds. And G will bind to C by 3 hydrogen bonds. You can also have reverse Watson-Crick. In reverse Watson-Crick, what happens if you imagine this dotted line and you flip, if you flip this second residue along the dotted line, so the instead of this carbonyl is now participating in the first, let us call this carbonyl 1 was participating in the bonding. Now carbonyl 2 is participating in the bonding. That is what you are calling as, this is actually GC is, so this is not correct actually. Sorry, I'll just discard this. Right. So we can only compare this and this, sorry. In reverse Watson-Crick, if you imagine this line along here, it flips along this line. So initially carbonyl 1 was participating in the hydrogen bonding. Now carbonyl 2 is participating in the hydrogen bonding. But in Watson-Crick, the pairing is maintained. A goes with T or U and G goes with C. In case of Hoogsteen also, that rule is maintained. A goes with T or U and G goes with C. But the difference in Hoogsteen is that in case of Watson-Crick, from the purine, only the 6-membered ring was participating in hydrogen bonding. In case of Hoogsteen, one NH from 6-membered ring and one from 5-membered ring participates in the hydrogen bonding. So both 6 and 5-membered ring participate in hydrogen bonding in Hoogsteen base pairing. Wobble is the case where the pairing changes. So instead of A with U, G goes with U, and instead of uh, uh, so A is we know that both Watson Crick and Hoogsteen tells you A should go with P or U and G should go with C. Now, if there is a mix up, A goes with C, that is basically wobble, A going with C, or G going with C, G going with U, that is also wobble. You can see G going with U is wobble. So the answer saying guanine binds to uracil in wobble base pair, that is D. Now the question is asking the fine structure of the 1' 2' cross peak in the C2' endo sugar geometry has how many components? So we know that in COSY, C2' will interact with, you can see C2', so C2' will interact with C3, you can see this C2' will interact with C3', it will interact with C1', then C2 double prime will also interact with C1'. So basically this is COSY, so we are only looking at adjacent hydrogens. So here we are talking about the 1', 2' cross, 1', 2 double prime cross peak. So it is talking about this cross peak. What will the fine structure of this cross peak look like? So we will come 1', 2 double prime. This is the one they are talking about. So 1' will be split by, for this we need to know the relative magnitudes of the splitting. So we can remember this, that for C2' endo, these are the relative magnitudes of the J. So now we will look at the fine structure. 1' is split by, we are looking at 1', 2 double prime. So this 1', 2 double prime is your active coupling. Active coupling, you can see 1', 2 double prime active coupling, and that will result in change of phase. So it will divide into plus minus. Then you have 1', 2'. 1', 2' is passive coupling. So plus will give plus and minus will give minus. And because the passive coupling is 10 Hertz, here this crossing is happening. So 1' is splitting into plus minus plus minus. You can see here. As for 2 double prime is first split by 1', that is the active coupling of around 6 Hertz. And because it's active coupling, it is plus minus. Then 2 double prime can be split by 2', and because this is passive coupling, plus splits into plus, minus splits into minus. And this 2 double prime, 2' geminal coupling is very strong. It is 14 Hertz. So that is why again this crossing is happening. It is plus minus plus minus. You can see that. There is another splitting of 2 double prime with 3', but that is very small. So it is ignored. You can see 2 double prime, 3' is only 1 to 2 Hertz. This is not observable, it's ignored. So you have 4 components on either side, and 4 into 4 gives you 16 components. So the correct answer is B, that is 16. The next question is saying, replacing 1 or 2 oxygen in the phosphodiester bond with sulfur in DNA results in what? How will it affect the melting temperature? So let us look at this case. The phosphodiester bond is this P double bond O and O on either side connected to the 5-membered sugar ring. Now, instead of this double bonded O, if you make it double bonded S, that is called a phosphothioate bond. It can be a phosphomonothionate where it is, so the unmodified is just written as with the nucleotide and the pentose sugar written as small p. Now, the one oxygen being replaced by sulfur is written as CPS, GPS, and so on. So one S is written. And if both oxygen are replaced by sulfur, phosphorodithioate, then it is written as CP2S, GP2S, and so on. As the oxygen is replaced by sulfur, the DNA becomes more unstable. So the melting temperature becomes lower. That is best seen in this graph. So you have temperature versus absorbance. The unmodified DNA shows a melting temperature around 60 degrees Celsius. When a single thiol is added, melting temperature becomes around 50. And when 2 sulfur is added, melting temperature becomes around 22. So with the addition of sulfur, the DNA is getting destabilized and the melting temperature is decreasing. So it is lowering of melting temperature, that is option B. The next question is asking, which among the following residues does not have side chain proton resonances in the range 6.5 ppm to 8 ppm? So if we remember the range of the 1H NMR chemical shifts of the amino acid, 6.5 to 8 ppm is basically this region. 6.5, we can take it. So any amino acid that has an aromatic side chain or a amide hydrogen in the side chain will have resonances in this range. So phenylalanine will have, because of the aromatic side chain, there is resonance in this range, so it is out. Asparagine has a side chain NH2, CONH2 group. Because of this, it is also out. And histidine has both. It has side chain NH and it has an aromatic side chain. So histidine is also out. So valine is the only one which will not show anything in this range. So valine is the correct answer, that is C. The next question is asking, in the Tandy algorithm for geometry optimization, which of the following are required? So let us briefly look into the Tandy algorithm. Tandy stands for torsion angle based distance geometry. And basically we start from many different initial structure. So here we are starting from 6 initial structure, 1, 2, 3, 4, 5, and 6. We do this because we don't want the calculation to be biased by the choice of initial structure. If all the initial structure leads to the same common final energy optimized structure, then we know that the optimization is correct. So we optimize each initial structure after putting the distance and torsion angles obtained from NMR. So we know that from NOESY we can get distance constraints, that NOE directly proportional to 1 by r to the power 6. So we will get distance constraints. We can get torsion constraints from Karplus equation and so on. So we will put all these constraints and then optimize each initial structure, and we will continue the optimization till all the constraints are satisfied. If all the final structures are same, that is, we get a unique structure no matter what we start with, then we will know that our constraints are correct. So in order for this algorithm, what is needed? We need multiple initial structures. So A is needed. Distance constraint is needed. Torsional angle constraint is also needed. So the correct answer is D, that is all of the above. Next, the question asks, the change in cargo trafficking with pH in DLC8 protein can be studied by what? So DLC8 is basically the dynein light chain protein. What happens is that if you imagine a tube and this is the protein. So the protein basically attaches the cargo molecule on one side. So when it is a monomer at pH 3, it remains as the native protein. At pH 7, it becomes a dimer. And when it is a dimer, it is able to connect to this cargo molecule and carry it from one end to the other. So how can this be studied? Basically, we can do pH dependent HSQC NMR. So this is 1H on the x-axis, 15N on the y-axis. At pH 7, the protein NMR, each of the peak will be at different positions. So this is glycine 63, this is serine 64, so on. When we add the cargo peptide, there is a change in the chemical shift. So glycine is shifting here, serine is shifting here, aspartic acid is shifting here, and so on. Now, if we reduce the pH to 6, we can see that some of the peaks are shifting back to the same position that they occupied in the initial case, that is, in the absence of cargo. So we can see that glycine and serine maintain their peaks, but isoleucine and aspartic acid, they are going back to the original position of the native protein, which it occupied in absence of this cargo. So at lower pH, the efficiency of binding of the cargo is reduced because the NMR is showing peaks that correspond to both peptide plus cargo and only peptide. So that means all the peptide molecules are not binding to the cargo because the binding efficiency is reduced by lowering the pH. So basically, we can study this by using this kind of pH dependent HSQC spectra. So the correct answer is option B, that is HSQC. Just give me a couple of minutes. Right. So the question is saying which experiment is helpful in studying large protein-protein complex where some signals are disappearing after binding. So straight away we can leave out COSY and TOXY because they are not good for studying large protein-protein complex. TROSY is an experiment we know we can use to study large protein-protein complex. What is basically done is that in HSQC, basically if you have a 1H15N HSQC and due to the NH coupling, they will split into 4 peaks. What is done in TROSY is first it is decoupled, so you get only one central peak, and then using the TROSY pulse sequence, we are able to get only the sharpest component of this cross peak. So only this component is retained. If we retain all 4 components, what happens is that when there are so many amino acids in a large protein-protein complex, the components will start cancelling out. So instead, only the sharpest component is filtered out for TROSY. So TROSY is often used for studying large proteins. But in some cases, where binding happens. After binding, many of the TROZ signals disappear. So you can see this is TROZ for the free protein here, and when it is bound to SR1, most of the signals are disappearing. In this case, there is a special experiment called crypt TROZ, which retains all the peaks even on binding, and this can be used to study the association process. So TROZ gets cancelled out because the question itself is saying that signals disappear on binding. So the best experiment to study it is crypt TROZ. That is option D. Next, you have a question saying secondary chemical shifts for C alpha in alpha helix and beta sheet are what? So secondary chemical shift delta S is basically the difference in chemical shift that is observed minus the difference in chemical shift of a random coil peptide. What is seen that in alpha helix, this secondary chemical shift is in alpha helix, this secondary chemical shift for C alpha is positive, H alpha is negative, and CO is positive. And in beta sheet, the signs are opposite. C alpha and CO are negative, H alpha is positive. So using the secondary chemical shift, the secondary structure can be predicted. An example is shown here at 8 molar urea concentration where the protein is completely unfolded. You can see that here the H alpha is plotted. So at 8 molar urea concentration, most of the peaks are positive. So most of H alpha being positive indicates a beta sheet. So most of the residues have a propensity to form a beta sheet. As the urea concentration is decreasing, the positive values are reducing. So you can see the beta sheet propensity is reducing. Till at 3 molar urea, most of the residues have very small values of delta S. So there is no specific structural propensity at 3 molar urea. Basically, using this secondary chemical shift, you can predict the secondary structure. So the question is asking secondary chemical shift for C alpha in alpha helix and beta sheet. So we are looking at this column. C alpha in alpha helix is positive, beta sheet is negative. So positive and negative. B is the correct answer. The next question asks which of the following are true for fast exchange? So we look at the different spectra at the different exchange rates. At slow exchange, when the rate of exchange is much less than the frequency difference of the two peaks, two distinct peaks are seen both in 1D and 2D spectra. In intermediate exchange, where the exchange rate is more or less equal to the frequency difference, the peaks are just starting to merge. And even in 2D, you can see they are just starting to merge. And in fast exchange, where the exchange rate is much higher than the frequency difference of the two peaks, you will not be able to distinguish any peak. It will just be a blur like this, both in 1D and 2D spectra. So which of the following is true for fast exchange? This is false. You cannot see distinct peaks. This is true. Peaks are broadened and merged. What about the option C? In fast exchange, what is done is chemical shift in either of the two dimensions. So you have, suppose this is 15N, this is 1H. Net chemical shift is calculated by this formula, that is delta delta H. So the difference in the shift in the, so we can imagine this as delta delta H is this one, x-axis, delta delta H. And along y-axis, it is delta delta N. So delta delta H whole square plus 0.1 into delta delta N whole square root is giving you a combined shift. And where this 0.1 comes from? It comes from the ratio of the gyromagnetic constants of nitrogen and hydrogen. So it is saying a combined chemical shift is calculated after including the variance in chemical shift of hydrogen and nitrogen after fast exchange. This is true. So both B and C are true. So the correct answer is option D. Next, the question asks, which of the following statement is false for water log Z based determination of protein ligand binding? We will briefly look into the water log Z technique. It stands for water ligand observed via gradient spectroscopy. It is extensively used for the screening of protein ligands. It is a relatively high throughput method and it observes the ligand, so it is a reliable method to identify small molecules. And it can detect binding constants in the micromolar to millimolar range. So clearly the first option is wrong because it is saying that it detects in the nanomolar range, but actually it detects in the micro to millimolar range. Now what happens in water logs is that the water is irradiated. You can see this is irradiating the water molecules, and this magnetization is transferred to the protein, and from the protein it goes to the ligand, and then the ligand is observed. So this method, the basis of this method is the transfer of magnetization from water to protein to the ligand via the nuclear Overhauser effect or NOE and chemical exchange. In water log, see, basically in absence, suppose you have a spectra in absence of the ligand and in presence of the ligand. So the what is observed is that small molecules that do not bind protein gives positive NOE signals. So you can see this positive signal means it's not binding. Positive signal means it's not binding. And what is binding to protein gives negative NOE signal. So this is binding, this is binding. So if it's not binding, it gives positive signal. If it is binding, it gives negative signal. So you can see the other points. That is not binding gives positive signal is correct. That is binding gives negative signal is also correct. And point B, that it relies on the transfer of magnetization between water molecule, protein and small molecule via NOE and chemical exchange is also correct. And the question is asking what is false. So the only thing false is option A, that is saying the binding should be in the nanomolar range. When we saw that in water log C, the binding should actually be in the micromolar to millimolar range. We will look at another question now, that which of the following is true for in vitro and in vivo NMR. So we know that the advantages of NMR based study, whether it is in vitro or in vivo, is that it is high throughput, the resolution is high, we don't have to give much effort on sample preparation. So X-ray crystallography, we have to crystallize it, purify it. There's a lot of sample preparation. NMR requires minimum sample preparation. NMR also gives a very quantitative, accurate idea. And the NMR method is non-destructive. Just a weak magnetic field is applied. It does not destroy the sample. You can also detect all the metabolites simultaneously in a single analysis. Your entire spectra will give you peaks of all metabolites. So NMR gives structural and quantitative information. And if you use specific NMR like 19F or 31P, where these kind of molecules are not present in the body that much, then you will not have interference from the native molecules in the body. So there are several advantages of NMR, whether it's in vitro and in vivo. In vitro basically includes fluids like plasma, serum, urine, saliva, or other secretion. But the problem with in vitro is that it is indirectly measuring the concentration of the drug. So you're taking the blood out and measuring the concentration of the metabolite in the blood, but it is not measuring the drug level at the exact site at which the drug is interacting. That is where the in vivo NMR comes in. In vivo, the advantage is that it measures the concentration of drug in the exact location where the drug is acting, and this is helpful because the plasma is just an indirect measurement. So now if we look at the statements, which of the following statements is true? All metabolites are detected simultaneously in a single analysis in vitro and in vivo NMR. This is true. In one spectra, you will see all the metabolites. In vivo signals originate only from labeled probes. This is false, because suppose you are doing 1H NMR. 1H you will get from the drug, but you will also, there is so much water present in the body, so many lipids, carbohydrates, and so on that have 1H. So you will have interfering signals from native molecules present in the body. This can only be prevented if you do a special NMR, you can see here, like 19F or 31P, where you don't have these probes in the body. And the third is that in vivo NMR allows us to measure the metabolite level in situ. This is also true because it is measuring it exactly at the place where the drug is acting. So the true statement is both A and C, that is option D. Just give me a couple of minutes. Right. The question is saying, which one of the following NMR assisted drug discovery methods requires the assessment of a small and diverse library of low molecular weight ligands? So the method is called shape screening. Basically, you have a small library of low molecular weight ligands and you screen them against the drug target. So, and the NMR is ligand based. So you're observing the ligand chemical shifts. So these initial guesses are derived from commonly known drugs. Initially, you will get only weak binding hits like millimolar range. But as you, so this is the initial guess. Now the guess can be improved and you can see we can get micromolar binding. And then on the basis of this initial finding, a library of analogues with further derivatization of the initial core structure was screened to obtain higher affinity ligands. So from micromolar, further derivatization can improve even up to nanomolar. So this kind of shape screening has been used as a starting platform for virtual screening of compound analogues to guide medicinal chemistry. So shape screening, so the basics is that using ligands that are known in common drugs, it is started and we get initial weak binding ligands, and after improving and derivatizing them, we can improve the binding. And we are observing the ligand NMR chemical shifts. So the correct answer that a small and diverse library of ligands is screened is shapes NMR, that is C. Next question is asking which of the following statements regarding relaxation data interpretation is true. So we know that if a protein has both rigid section and flexible section, then the longitudinal relaxation rate, or R1, is high in the flexible region. So this is the flexible N terminal, flexible C terminal. So R1, longitudinal relaxation rate, is high in the flexible region, that is N and C terminal, and it is low in the rigid region. this inner core. R2 is exactly opposite. It is low in the flexible region, N and C terminal, and it is high in the rigid region, that is this inner core. If urea is there, basically the structure is completely uh, denatured, and so uh, all the R2 more or less have equal values. So this is what urea causes. It causes this difference in R2 is completely lost, and all of them have similar and low values. And inside R2, there is something called R exchange, which basically gives you idea about exchange phenomena that is happening in millisecond to microsecond time scale. So if you calculate R2 in different concentrations of urea and you take a difference, so delta R2 is R2 in 8 molar urea minus R2 in 7 molar urea. This one is delta R2 7 molar minus 5 molar. So this delta R2, what it will do, it will cancel out the intrinsic R2 and it will give you information about the contribution of this exchange phenomena. So this is about how R1 and R2 is telling you about the dynamics of the protein. So if you look at the question, flexible region of protein have high R1. That is true. R1 is high in flexible region of protein. So this is true. Flexible region of protein have high R2 is wrong because R2 is low. Urea does not affect relaxation rate is wrong. We just saw urea is affecting the relaxation rate. And R exchange affects R1 is wrong because R exchange is affecting R2, that is the transverse relaxation rate. So the correct answer is option A. Next, the question is asking, the relaxation rate of protein is primarily affected by what? So we know two factors contribute to the relaxation rate. One is the dipolar relaxation. So if you have two spins, the dipolar interaction between them is defined by m0 by 4 pi into h bar into the product of the gyro-magnetic ratio divided by the distance between the two spins cube, and in the bracket, the angular component, the one which is defining the magic angle of spinning, is 3 cos square theta minus 1, where theta is the angle of the spin with the B0. In magic angle spinning, we consider this to be 0, which is where theta comes out to be 54.7 degrees. So dipolar interaction is responsible for the relaxation phenomena. And another contribution comes from the chemical shift anisotropy. So basically, suppose you have the tumbling of the molecule is very fast, like 15,000 hertz. So chemical shift anisotropy gets averaged out and you get a single sharp peak. This is what happens in solution state NMR. But in solid state NMR, where the tumbling is very slow or it is completely restricted, then each orientation will give a different chemical shift. And what you will observe is a net envelope of chemical shifts. So the solid state NMR is very broad. This is called chemical shift anisotropy. So these are the two phenomena mainly responsible for relaxation rates. So you can see A is the dipolar interaction, B is the chemical shift anisotropy. So the correct answer is D, that is both A and B. So that brings us to the end of the revision session, which is basically covering all the so many of the topics from the first to the last class of this course. And so this has kind of been like a revision session where many problems have been solved and many concepts have been discussed. So as always, I will upload this with the slides and the video, and you can go through it while revising for your exams. I will end the class now.